Hello, everyone, and welcome to today's live broadcast, Engineering Stem Cell Biology for Disease, Modeling and Therapeutics, presented by Amr Abdeen. He's a postdoctoral associate at University of Wisconsin-Madison. I am Marjorie Torres of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your questions into the box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Please join me in welcoming Dr. Amr Abdeen. I will now turn the presentation over to him.
Good morning, everyone. My name is Amr Abdeen, and I'm a postdoctoral researcher at the University of Wisconsin-Madison. Today I'll be talking about engineering stem cell biology for disease modeling and therapeutics. I work in the University of Wisconsin-Madison at the Wisconsin Institute for Discovery. I work in Chris Saha's lab, where we work on engineering of stem cells. So it's a very exciting field, and I'm going to walk you through some of what's happening today. So stem cells, they're defined by two key properties. They're multipotent. That means they're able to differentiate into multiple cell lines. And they're self-renewing, so they can propagate in culture indefinitely. And there's kind of two main categories of stem cells. There's embryonic stem cells, and these are isolated from blastocysts, which is a stage in embryogenesis. And so to get these embryonic stem cells, they're isolated from this inner cell mass in the blastocyst. And what's special about these cells is that they're pluripotent, meaning they can differentiate into virtually any cell in the body. They're kind of naive cells that form the embryo and that later differentiate into all these different cell lines. So they can make skin, they can make muscle, they can make neurons. One problem with them, though, is that there's sometimes ethical objection to obtaining or using these stem cells. Another cell type that's less controversial is adult stem cells, and these exist in a variety of tissue. They are multipotent, so they can differentiate into some subset of cells in the body where they act as a support and repair system. So, for example, you have stem cells in the blood, which generate new blood cells and help, repair injury. You have stem cells in the brain, you have stem cells in the gut, and you have stem cells in the skin. One advantage of using these cells is that you can use them autologously, which means you can get cells from the patient and then transplant them back into the patient for a therapeutic purpose. And this is good because you don't need to find a match from some another patient, and you can extend these cells ex vivo before transplanting them back in. And so these stem cells, there's been lots of advances in the last few years, and people have figured out lots of very clever ways to manipulate them for many purposes. So I'm going to go through some stem cell engineering tools that we use. Namely, I'll talk about reprogramming and differentiation of stem cells. I'll talk about genomic perturbation or working with the genomes of these stem cells. And then, although there's multiple bioengineering strategies, I'm going to focus on two that are particularly relevant for disease modeling and therapeutics. So reprogramming is actually a very, well, it's relatively new now, it's been over 10 years. But it was a very striking discovery. So people used to think that these embryonic stem cells will, it's a one-way differentiation from the embryonic state to this differentiated cells. So once a cell is differentiated or specialized into, say, a fibroblast or a skin cell, it can go back to this pluripotent state. But what Shinya Manaka discovered, and he got the Nobel Prize for this, was that by activating 4 genes, so OCT4, SOX2, KLF4, and c-MYC, he was able to get fibroblasts from skin biopsies to turn into what he called induced pluripotent stem cells, which are cells that are, they look like embryonic stem cells. They can differentiate into the 3 different germ layers, so they can basically differentiate into all the cells in the body. And you can culture them in vitro indefinitely as well. And this is great, so it acts as a cell source. So when you're an adult, you don't have your embryonic cells anymore, right? So what you can do is you can get somatic cells which are already differentiated, and using this reprogramming process, you can get pluripotent stem cells and use these as a cell source for therapies, for example, or for modeling a disease, or for transplantation back. So you can take cells from a patient and transplant them back into the same patient, again, foregoing the need for a match. And so these cells are now becoming a very big resource that for tissue engineering and therapeutic purposes. Another thing is, so once you have stem cells, you need to be able to control how they differentiate. So once you have iPS cells, which you can see in the middle here, they can differentiate into down multiple pathways. So depending on which pathway they go, they could end up being a neuron, or they could be like a cardiomyocyte, a beating heart cell. So it's very important to be able to control that process. And controlling that is often done by using guidance from developmental biology, seeing how these embryonic cells in the embryo develop and are specified into different cell types or tissues in the body. And a lot of the time it ends up being trying to emulate that process, as you can see through this example. So on the right you can see a part of the spinal column development, and there are different neuromesoderm progenitors there. Progenitors for the hindbrain, for the lumbar part, for the thoracic part. And so how cells specify here depends on a host of different transcription factors. And by studying how this development happens and what kind of factors turn on at what time, people have been able to figure out how to get an embryonic stem cell or a pluripotent stem cell to a specific lineage that they desire. So it's still ongoing, but people have developed some very efficient differentiation protocols to be able to get these stem cells into the cell type that they need. So reprogramming differentiation basically gives us this very large source of cells that we can kind of control how they differentiate, and we can make different tissues or different cell types of these cells, of them. Another thing we can do is we can manipulate the genomics of the cells. So this is not really new. So people have done this for a while now, where you can use nucleases to make cut in the cellular DNA, and then you can use that cut to make changes in the DNA sequence of the cells. So what people were using before, where these are still using, is nucleases based on protein-DNA interactions. So what that means is you have these proteins that can recognize a specific sequence of DNA, and by chaining them together, you can identify some specific point at the DNA and then they attach a nuclease domain, a cutting domain. And that cutting domain allows you to make an edit at the DNA at that specific point. The problem here is you have to engineer a different protein for every time you need to target something different. So let's say you develop a protein to target one gene, but then to target another gene, you have to engineer that protein again for that different sequence. A lot of you probably heard about CRISPR, which is a recent technology. And so what's really nice about CRISPR-Cas9 is it's much easier to design and use. So Cas9, the advantage is it's an RNA-directed or RNA-guided nuclease. And what that means, in order to target a specific sequence of DNA, in order to get a single match, the guide is about 20 nucleotides. And what that is a 20 nucleotide sequence that's complementary to the DNA. So basically, by a very simple synthesis of an RNA strand that has these 20 nucleotides coding where you want to target, you can target anywhere. So if you have the sequence for a gene, pretty much you can find, within a few days, find a guide to cut there, as opposed to weeks or months with previous nucleases. And so far I've just talked about cutting. So what happens after you cut the DNA? So here you see on the left you have this Cas9 nuclease. It's guided by this sgRNA to this spot in the genome. And when it attaches to that spot, it causes a double-stranded break in the DNA. So the DNA strands are pulled apart, or cut apart. And then one of two things can happen, basically. You can have the non-homologous end joining. And what that means is the cell tries to repair the DNA by inserting random bases where the cut happened until it's repaired. And what happens there is you get insertions or deletions of the DNA sequence there, which can mess up the gene, so it can knock out the gene, or it can cause some change in how the protein is formed from that gene. The problem is this is not a controlled process. So when you induce that cut, it's random basically what happens at that cut site. So a more precise way to edit is shown at the bottom, which is called homology-directed repair. And with that, we have targeted sequence replacement. So basically, in addition to the Cas9 and guide RNA, you add a donor DNA, encoding what you want to insert at the cut. So it could be a different sequence, it could be a whole new gene, it could be a specific mutation you want corrected or edited. And what happens there is that the cell can actually work with the template and repair the DNA according to the template that you give it, effectively giving you the ability to modify the genome of the cell. And that comes in really, really handy, as you'll see later. So CRISPR hasn't only been used for cutting or modifying the genome. It's been engineered, so there's been lots of engineering going on to be able to use it for other purposes. And basically what they take advantage of is the ability of the Cas9 enzyme to bind to the genome at a specific spot. So here in part A, what you see is a dCas9, which is deactivated Cas9. And what that does is it binds at that specific spot, but it does not cut the DNA. And while not very useful on its own, people have been able to append functionalities at dCas9 to be able to perform multiple functions at that spot. So for example, you can append a fluorescent dye to the dCas9 and be able to image DNA at that specific spot. So just find where sequence is inside the cell. What you see in part B is gene activation. So here you can add a transcriptional activation, which will turn on a gene, causing the cell to express it, even if it doesn't usually. And these are helpful for if you want to see the effect of a specific gene that's not expressed in a specific cell type, for example. You can also see transcriptional deactivation. So you can turn off the gene without knocking out the DNA. And that's helpful because, again, it's mostly helpful for studying how the cell behaves or the function of that gene, but it could be also used for in vivo therapeutics. With other functionality, this has made CRISPR a very versatile tool in the lab. And you can see there's lots of applications for it, so you can use it going from A to do knockouts of genes, so just study the effect of knocking out the gene. You can study the effect of knocking in transgenes or just different genes. You can study knock-in at different spots in the genome, see if transcription is affected that way. You can do mutagenesis screens, so you can introduce mutations and see whether the effects of these mutations. So the CRISPR systems have been adapted for RNA editing, so you can use them to edit the RNA in a cell. Again, gene silencing and activation. And a big component that's gaining in popularity is these CRISPR screens, where you can actually encode guide RNAs for all the genes, let's say 20,000 genes in a library, and transfect that library into a pooled group of cells. And by that, you can get a group of cells that express. So different cells have different knockouts, and you can do these screens to see if a specific subset gains some function. So you do a screen and have some selection pressure to see a specific function that you want, and that's a way to find genes of relevance, so to speak. And then, so in addition to genomic perturbation and reprogramming of cell types and differentiation, there's also lots of what I call bioengineering tools. So these are tools that, in addition to the other two, there are many. It's a very, very broad category, but they can be used to specify and further control how the cells behave. And the need for these tools partly comes from the complexity in the in situ environment. So on the middle here, you see a cell in its native environment. So it has specific geometry. It's in contact with other cells. There's a basal membrane, so there's an extracellular matrix. It changes. There's movement. There's specific mechanics. So in a live system, in vivo, the cells have a very maybe not specific, but a very defined environment. Well, when we generally do cell culture, as you see on the right, we generally just culture cells on plastic, which is nowhere near as much control as they see in vivo. And so this makes one of the more important bioengineering tools is synthetic scaffolds. And with that, we try to recapitulate some of the properties we see in vivo in the lab, so we can be able to control the cells in that same way. And there's lots of relevant properties. So when we make a scaffold, so hydrogels are a prominent example just because they can have the same kind of properties like human tissue sometimes. And what we find with these scaffolds is that the stiffness, for example, will really affect how the cell behaves. It will affect how the cell spreads. It will affect how the cell moves. And it will affect how stem cells differentiate. The composition of the matrix, so what kind of proteins are outside the cells, interacting with the cells, growth factors, how these, so ligand presentation is how these extracellular components are presented to the cells. So are there cryptic sites, or is the cell able to attach to specific moieties or not? And so in addition, you can control cell shape, cell topography, and you can control most of these factors in a dynamic fashion. Not perfectly, but it's rapidly improving. And by doing that, we're able to much better control cell behavior in a predictable way. So another thing about the cell that we saw in vitro was that we usually culture cells as separate cells, so not as a group of cells, and that's not always very helpful. So if you look on the left, you can see that the organization of the body, it gets increasingly complicated with scale, where you go from subcellular to cellular organization, and then you get sheets of cells, for example, and then you get tissue, and then you get organs, which are, so you add levels of complication as you increase with scale. And in life sciences, so what we're trying to do is rival that complexity. So instead of going from monolayer cell culture, we're trying to do culture cells in spheroids and then move on to organoids, to tissue explants, and maybe even more complicated. But right now, the state of the art is around the organoid area. And so how that works is, if you look at the right, so we're looking at the intestinal organoid. And looking at the in vivo intestine, you can study that, and people have found that there are these intestinal stem cells that regenerate a large part of these villi-like structures in the intestine. And by isolating these stem cells and studying their properties, people are able to culture them in vitro in a way that causes them to make these intestinal organoids, which actually have a lot of the properties in common with the in vivo intestine. So they do nutrient exchange, for example. And what these can be used for, presumably, is the modeling of diseases in the intestine, or they can be used, transplanted back if they are functional enough that they can cause a therapeutic effect. And again, here people will borrow from development. So we're looking at the brain organoid here, and by looking at how during development the brain develops different regions, we can model based on that to try to make these different regions in the lab on a dish. And by further engineering the environment to induce these regions in different spatial areas, for example, we can kind of recapitulate how the brain looks in vitro. And this is very useful. So going from single cells to tissues, there's very clearly many properties that will change and improve, and make it more difficult, if not impossible, to predict what will happen just by studying cells. So the organoids are a very exciting way to see that complexity in a dish without having to do in vivo studies. So there are caveats. So although you have a near physiological model system, and they can be cultured, and there's, if you look on the right, there's been organoids made from multiple tissues. There's been lung, liver, stomach, intestine, brain organoids. So these are all in development. However, they are complex and challenging to culture, and they're not in the complete native environment, so there are still differences. And there's an absence of other systems that interact with it in vivo. So for example, the immune system may not be represented in an organic culture, so you're not able to investigate the effects of inflammation, for example, in some of these systems. But they do remain a valuable tool. So using these tools, there's been a large improvement in our ability to model human diseases, especially using patient-derived cells. So looking back and trying to get a bigger picture here, we have a patient carrying a mutation that's causing a disease. What we can do is we can get somatic cells, so let's say a skin biopsy or a blood biopsy, reprogram these cells, and get pluripotent stem cells from the patient, which we can use to differentiate into different, for example, differentiate them into gut stem cells and form organoids from that. We can, so in multiple different organs. And having that disease model, it can be used to study how drugs will interact with the disease in vitro, so without testing in vivo, right? And we can do, we can check for biomarkers that we can use to detect specific disease phenotypes earlier, for example. And finally, we can do cell replacement therapies based on these models, where we can perhaps correct a mutation and transplant back healthy tissue. But modeling human disease is not, it depends on, it's context dependent. So if you have cells, there's only so much that you can model based on single cells. Some of the, again, the structure or architecture of the tissue might not be captured there. Even with organoids, some of the tissue interactions or the more complex interactions may not be captured in organoid culture. However, with organoids, you get much better, in some cases, you get much better control with some of the profiling you can do. So you can do large kind of host microbe interactions. You can monitor the effects of drugs in a structured tissue, for example. And you can see the effect on phenotypes that are not, you can't get with cells, such as uptake through alveoli, for example. So that makes modeling using these patient-derived stem cells, it's not suitable for anything, right? So there are some criteria that help when selecting a disease that you'd want to model with patient-derived stem cells. So the inheritance is the disease based on a single gene. It's easier to model than some of the polygenic diseases. Penetrance, which means if a disease has high penetrance, it would mean if you have the gene or a specific mutation, you have the disease. While if you have low penetrance, the relation is not that clear. So you could have a specific mutation and not have the disease or have it. So it's not a clear relation between genetics and disease phenotype. Developmental disorders are easier to model than late onset ones because of the nature of the system. And it's optimal to do this when you don't have a suitable animal model. So if you have a good animal model, it might not be very useful to try patient-derived disease models. And again, simpler phenotypes are easier to study using these systems. And accessible differentiation that's efficient and well-controlled would be better if you're trying to model a disease using stem cells. So I'll give an example that we've been working on, which is dystrophinopathy, and that's a degenerative eye disease. And so on the left, you can kind of see the lesions in the retina in the eye seen with that disease, and the bottom is kind of a simulation of someone who might have that disease, how their vision might look. And this is a disease of the RPE layer at the back of the eye, boxed in red here. And the RPE layer is the retinal pigment epithelium. And the retinal pigment epithelium is this layer at the back of the eye that supports the photoreceptors on the retina. And one of its main functions is to recycle byproducts from the retina. And when it fails to do that, there starts to become degeneration in the retinal layer and in vision. And this is a disease in a known gene, it's the BEST1 gene, which encodes for bestrophin 1. And bestrophin 1, as you can see on the right, is this transmembrane calcium-activated chloride channel. And so this is activated by calcium, and it's a chloride channel. And mutations in this gene can cause what's called BEST disease. So although it's a monogenic disease, it's in this single gene, um, there are other advantages to study this gene using a stem cell model. So it's a rare disease, and this is important because large diseases such as cancer, you can use these big data approaches where you can look at thousands of patients and look at gene-wide association studies and try to tell which genes are common for specific symptoms or specific diseases and do it that way. However, for diseases that are rare, what are called orphan diseases, it's hard to use that same approach with so few samples. So here it becomes important to be able to really control the model you're using to be able to get useful data. And so bestrophinopathy also has mechanistic similarities with other degenerative diseases in the macula. And it's an interesting disease just because, so what you're looking at on the left in this slide is an amino acid sequence of the protein, and colored residues represent disease-causing mutations. And you can see different mutations will cause different kind of phenotypes. And depending on whether your mutation is in the calcium class, boxed in blue, or the chloride binding areas boxed in pink, the disease can have different, maybe not phenotype, but different mechanisms. And so this makes it important to actually study not just the gene, but specific mutations, making it also really convenient to be able to introduce or remove mutations. And people have been using this, so they've figured out ways to make RPE layers, retinal pigment epithelium layers, similar to organoids in vitro. And these layers are actually the closest thing to the human model, so they're even closer than mouse models. And you can, so they're iPSC derived, so they're derived from human iPS cells. And what you can use these is you can study the effects of drugs. So on the right, you're seeing a study where one of the jobs of the RPE layer is to recycle photoreceptor outer segments. But when you have a defect, these photoreceptor outer segments accumulate, causing degeneration. And so what you can use these models for is you can study the effects of drugs, for example. So some drugs may work for a specific mutation, but they may not work for others. And that gives it a personalized medicine screening dimension, where you can take the cells from a specific patient, study the interaction of drugs for this patient, and hopefully get a more specific idea of which drugs to use on a patient-to-patient basis. So this is already pretty complicated, but I wanted to see what we can do with other diseases that are not monogenic, so polygenic diseases. And here I'll talk about Parkinson's disease, and two main mutations or genes involved in Parkinson's disease is the leucine-rich repeat kinase 2, LRRK2, shown in red here, and synuclein, SNCA, shown in orange here. And if you get patients with Parkinson's disease and get cells from them, reprogram them, so you have these iPSCs, or induced pluripotent stem cells from the patients, what you can do is you can, using CRISPR or some gene editing method, remove or correct a mutation in one of the genes. And what this tells you is how much of the phenotype is caused by that specific mutation. And then you could get the cells, the original cells again, and correct the other one, and again ask how much of the phenotype is modulated by that mutation. However, this doesn't really capture synergistic effects, so to speak, especially if they're developmental. So here what we could be able to do is, for a healthy individual, get healthy cells that don't have the Parkinson's disease phenotype and introduce these mutations. And by introducing mutation in one gene or the other gene, you can ask, is this mutation alone, what's the effect of this mutation alone? And then by combining them, you can combine them in different sequences. You can study the synergistic effects that they have together. Again, it gets pretty complicated pretty fast, especially with diseases that have multiple mutations. And so it's not only whether this mutation causes this or that mutation causes that. So for polygenic disorders, you have these different modes that could happen. So it could be that one mutation causes something, another mutation causes something completely different, which is depicted on the top here. While it could be that mutations at different time points or at different stages accumulate to cause a specific phenotype, which would be, again, a harder model, a harder thing to model. But it's not just for polygenic disorders. So even for monogenic disorders, where you have one gene, it's not as clear as just removing the mutation and seeing if the effect goes or not. So for example, here on the left, you can see a patient A, and to study one mutation that might be causing a specific phenotype, we could correct that and see what happens to that phenotypic trait. And let's say we see a reduction as we see here for that circle bar, but then we get a patient B in green, and patient B, we do the exact same thing, we correct the mutation, but this change is not, it's there, but it's not as profound as patient A. So here it's not just the effect of the gene, it's just there's an influence of the background genetics or the epigenetics, or it could be a lifestyle-based effect. So it's not always that clear how the interactions happen. And on the right, so if you also try to introduce the disease into wild-type cells, patient A who had less severe symptoms to start with, he might, getting that mutation and introducing it into a control line might cause a more, a harsher effect than getting patient B's mutation, although patient B has more severe effects. And again, that has to do with the background genetics or how these cells, epigenetic changes in the cells. So the modeling is not very straightforward, but it's a very useful tool to be able to tell the influence of specific genes, drugs, in a much more sophisticated way than people will do with just single cells or patient-derived cells. So the ability to edit the genes, to produce specific mutations, or to control, to either correct patient cell lines or introduce disease into control cell lines is a very helpful mechanistic tool, in addition to the screening aspect. What you can also do with these cells is a therapeutic kind of direction. And stem cells have multiple properties that make them suitable for therapeutic use. So here I'll give an example of mesenchymal stem cells, which are found in the bone marrow, they're found in adipose tissue, they're found in multiple places. But these cells have multiple effects. So they have paracrine effects. They have paracrine effects, whereby they secrete growth factors, for example, that help angiogenesis. So they're frequently used for that purpose, where they're injected into an infarcted heart, for example, and they stimulate angiogenesis in the infarcted area. They have immunomodulatory effects, as you can see on the right, which help modulate the immune system, which can be desirable in some cases. And so these mesenchymal stem cells, they can differentiate down variable pathways. So they can differentiate into adipocytes, which are fat cells, chondrocytes, the cartilage. They can differentiate into osteocytes or bone tissue, basically. And by trying to control their differentiation, they're being tested for transplantation for degenerative diseases which cause loss in some of these tissues. And actually, there's several clinical trials using these mesenchymal stem cells. And as you can see, they're in a bunch of diseases. So they're in bone and cartilage diseases, neurological diseases, and cardiovascular diseases make up the bulk of clinical trials where MSCs are based. And for their immune modulatory properties, they're also in trials for graft versus host disease. So really being able to make use of these cells, controlling them, makes this large space where you can use them for different therapeutic purposes. And the advantage of mesenchymal stem cells is, so they're adult stem cells, there's no ethical concerns really. You can expand them ex vivo. And transplanting them, they're autologous, so you don't really need other donors. However, iPSCs, while they have some other disadvantages, they're also currently in clinical trials for several diseases. They're actually being transplanted in the eye for age-related macular degeneration. So this is actually RPE layers that we talked about in the eye, and they make the layers and they're transplanted into the eye. Dopaminergic neurons are made from iPSCs for Parkinson's disease, being tested for spinal cord injury, for making islet cells for diabetes, and for infarctions. So iPSC-derived cardiomyocytes are a very hot topic right now. So to summarize, I hope I convinced you that stem cells, whether they be adult, embryonic, or induced pluripotent stem cells, are a valuable resource for both modeling of diseases and therapeutic purposes. The ability to differentiate the cells, reprogram somatic cells into these pluripotent stem cells, edit the genes, or modulate the genome, and combined with other biomaterial strategies can be used so we can control the stem cell, or we're in the process of controlling stem cell behavior, their genotype, aspects of their phenotype, as well as the tissue level structure and organization. And so right now, stem cell disease models and therapies, they're advancing rapidly, and there's multiple therapies in clinical trials currently. And this has actually caught the attention of the FDA, which has designated a regenerative medicine advanced therapies, or regenerative advanced therapies designation, for an accelerated clinical testing and trial process. And with this, I'd like to thank you and take any questions.
Thank you, Dr. Amr Abdeen, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your question into the box that appears on your screen and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, can gene editing eliminate genetic diseases composed of single mutations?
Well, this is a complicated question because while theoretically gene editing does, so if you correct a mutation, it's no longer there. In vivo, it's a very complicated question. So let's say you have a genetic disease in a specific organ, right now we're not able to correct the mutation. So if you put in the Cas9 enzyme, you're not going to be able to correct it in all the cells. You're just going to correct it in a subset of the cells. And whether that helps or not, it's very dependent on the system in question. So for sickle cell anemia, for example, if you try to target the stem cells that make the mutated red blood cells, so this mutation causes the red blood cells to be not able to efficiently carry oxygen, causing that anemic phenotype. So if you try to target those stem cells forming the phenotype, and you correct the mutation in a subset of them, a subset of the red blood cells can form normally, and that will give a phenotypic improvement. Maybe not a complete improvement, but people have seen improvement in this disease in mice. However, if you think about a developmental disease, for example, if you edit the genes after some of the effects have happened, you may not be able to reverse that. One thing that people are excited about right now is germline editing, or editing in the embryo stage where all the cells in the body would be corrected, for example. However, it's not clear if that can be done efficiently right now. And it can. And there are ethical concerns to go with there.
Thank you for that answer. Our next question is, are there safety concerns from errant differentiation of pluripotent stem cells once transplanted?
Yes, so actually the ability of the pluripotent stem cells to differentiate can backfire in this kind of application. So it is a safety concern because stem cells will form teratomas in mice when implanted, and that just goes like kind of random differentiation of the stem cells, and it's not very safe. So typically what people will do is try to use the stem cells ex vivo, take the stem cells out or reprogram them, and then form a differentiated cell type and transplant that back in. So you don't run the possibility of errant differentiation. And there's usually very stringent selection and quality control that needs to go into ex vivo processing before transplantation back into the body to prevent these kinds of effects.
I would like to once again thank Dr. Amr Abdeen for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December of 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who might have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.